Eat a sandwich right now. Just picture it. Bread, turkey, a little mayonnaise. Take the bite, chew it, swallow it. Somewhere around 90 minutes from now, every molecule of that sandwich that makes it through your intestinal wall is going to pass through one organ before it touches almost anything else in your body. Not your bloodstream in general, one specific vein, feeding one specific filter, sitting just under your right ribs, weighing about 1.5 kilograms, doing something to that sandwich that literally nothing else inside you is built to do. That organ is your liver. And the reason it gets first contact with your food, before your heart, your muscles, or your brain ever see a trace of it, comes down to something called the hepatic portal system. And once you understand it, you realize it might be the strangest piece of plumbing in the entire human body. Here's how it actually works. Blood leaving your stomach and intestines doesn't head straight back to your heart the way blood from your arm or your leg does. Instead, it drains into the portal vein, and the portal vein dumps it directly into the liver, about 1.5 liters of blood every single minute, which works out to roughly 75% of everything the liver receives, and all of it arriving already loaded with whatever you just ate. Glucose, amino acids, fat packaged up as chylomicrons, alcohol if you had a drink with lunch, any medication you swallowed. The liver sees it first, and seeing it first means controlling it first. Pharmacologists call this the first pass effect, and it's the reason a pill you swallow and the exact same drug injected into your arm can behave completely differently once they're in you, because the swallowed version has to get past an organ that's actively trying to break it down before it ever reaches the rest of your circulation. Once that blood arrives, it meets something like 240 billion cells called hepatocytes, packed into millions of tiny hexagonal processing units called lobules. Blood pouring in around the edges, draining out through a central vein in the middle. And the first thing those hepatocytes do to your sandwich is sort the sugar. Your blood glucose has to stay locked inside a narrow window, somewhere around 70 to 100 milligrams per deciliter, because too much of it starts damaging the walls of your blood vessels through a process called glycation, and too little of it starves your brain, which burns through about 120 grams of glucose a day and can't store a single gram of its own. So the second that sandwich floods your portal vein with sugar, your pancreas releases insulin, and your liver, before your muscles even get a look, starts pulling glucose straight out of the blood and stringing it together into a branched storage molecule called glycogen. An enzyme called glycogen synthase does the actual stringing one glucose unit at a time, and your liver can bank about 100 grams of it this way, roughly 400 calories held in reserve. Then, hours later, once you haven't eaten in a while and your blood sugar starts sliding, the whole thing runs in reverse. Glycogen phosphorylase breaks that chain back apart, glucose spills back into your blood, and if the reserve ever runs dry completely, your liver does something almost unbelievable. It builds brand new glucose from scratch, out of amino acids and lactate, through a pathway called gluconeogenesis. Your liver is manufacturing sugar from protein, while you're asleep and not thinking about any of it. But sorting fuel is only the first thing happening to that sandwich. There's a second mechanism running at the exact same time, and this one has nothing to do with energy. It has to do with poison. The turkey in your sandwich was protein. Protein is built out of amino acids, and every single amino acid carries a nitrogen-containing amino group. When your body breaks those amino acids apart for energy or to salvage the spare parts, that nitrogen gets stripped off and it turns into ammonia. And ammonia, even at the levels a normal diet produces, is neurotoxic. It crosses the blood-brain barrier and disrupts neurotransmitters cycling badly enough that people in severe liver failure can develop something called hepatic encephalopathy, confusion, tremor, in the worst cases coma, purely because the liver has stopped clearing ammonia out of the blood. In a healthy liver, none of that ever becomes a problem, and the reason goes back to a discovery made in 1932 by Hans Krebs, working with a medical student named Kurt Henseleit in a lab in Freiburg, Germany, using thin slices of rat and pigeon liver tissue. What they found was that liver cells were converting ammonia into a far less dangerous compound, urea, through a cycle of 5 separate enzymatic reactions, a pathway now known as the Krebs-Henselite cycle, or just the urea cycle. 2 nitrogen atoms pulled from ammonia, 1 molecule of carbon dioxide, go in. 1 molecule of urea comes out, ready to travel to your kidneys and leave your body the next time you go to the bathroom. Your liver runs this cycle constantly, turning a genuinely toxic byproduct of eating protein into something your kidneys can discard without a second thought. And it does it so smoothly that most people never notice it happening, not once across a lifetime of eating meat and eggs and beans and cheese. So already, in the time it takes to digest one sandwich, your liver has sorted the sugar, banked what it could, released what your brain actually needed, and neutralized the nitrogen waste left over from the protein. And there's a third process running in that exact same window. This one's for the mayonnaise. Fat doesn't dissolve in blood. That's a problem, because blood is water-based. So your liver manufactures bile, somewhere between 800 and 1,000 milliliters of it a day, built from cholesterol, concentrated and stored in your gallbladder, then released into your small intestine specifically to break large fat globules into microscopic droplets, small enough for digestive enzymes to actually work on. But the liver's job with fat doesn't stop at digestion. Once fatty acids get absorbed and shipped back to the liver, hepatocytes package them up, along with cholesterol and protein, into transport particles called very low-density lipoproteins, or VLDL, and send them back out into the bloodstream for tissues that need the energy, or for storage in fat cells down the line. Your liver is essentially running your body's entire fat distribution network, deciding in real time what gets burned, what gets shipped, and what gets tucked away. There's a fourth mechanism at work here too, and this one has nothing to do with nutrients your body wants. It's about the stuff your body doesn't want. Even a plain sandwich carries trace plant compounds, preservatives, whatever medication you might have taken with it, and your liver processes every bit of that through a 2-step system built around a family of enzymes called cytochrome P450, more than 50 different versions of them, each one tuned to recognize a different class of molecule. In what biochemists call phase 1 metabolism, these enzymes chemically alter the foreign compound, usually by tacking on a reactive oxygen-containing group, which, strangely enough, doesn't always make it safer. Sometimes it makes it more reactive, not less. That's exactly why phase 2 exists. A separate set of enzymes attaches a water-soluble tag onto that altered molecule, glucuronic acid, sulfate, or an amino acid, turning it into something your kidneys can finally recognize and flush out. And this 2-step system is precisely why grapefruit juice can be dangerous with certain medications. Compounds in grapefruit block specific cytochrome P450 enzymes, so drugs that were supposed to get broken down on their first pass through the liver instead flood straight into your circulation at full strength, sometimes at several times the intended dose. Now here's where these 4 mechanisms stop looking like 4 separate jobs and start looking like one coordinated system. Your liver isn't just processing sugar, or just neutralizing nitrogen, or just packaging fat, or just disarming foreign molecules. It's running all 4 of those at once, on the same blood, in real time, constantly adjusting the balance between them based on hormonal signals. Insulin after a meal pushing everything toward storage, glucagon between meals pushing it back toward release, cortisol during stress cranking gluconeogenesis up even further. The liver isn't really a single-function filter. It's closer to a central metabolic switchboard deciding, cell by cell, minute by minute, whether the molecules coming in from your gut get burned, stored, exported, tagged for detox, or destroyed outright. And there's a 5th layer to all this, and it has nothing to do with nutrients at all. Scattered through the liver's blood-filled channels are specialized immune cells called Kupffer cells, the largest population of resident macrophages anywhere in the human body. Because the portal vein carries blood straight out of your intestines, and your intestines are packed with bacteria, that blood arrives at the liver carrying bacterial fragments, endotoxins, and occasionally a whole bacterium that slipped through the gut wall. Kupffer cells sit inside the liver's sinusoids and physically filter that blood, engulfing bacteria and debris before any of it can reach the rest of your circulation. Estimates suggest your liver clears the majority of gut-derived bacterial endotoxin out of your bloodstream on every single pass. Most people never learn this mechanism exists at all, and it means your liver is simultaneously running a chemistry lab and standing guard as your bloodstream's first line of immune defense. Filtering biology, not just molecules. And none of it happens through some single organ working in isolation. It happens because of an entire architecture built for exposure. Every hepatocyte sits directly against a sinusoid, a specialized capillary with gaps in its lining wide enough for entire proteins and lipoprotein particles to pass straight through. That's why the liver can sense and respond to blood chemistry faster than almost any other tissue you have. No basement membrane to cross, no tight junctions to squeeze past. The blood essentially bathes the hepatocyte directly. And that direct contact is exactly what lets the sugar sorting, the nitrogen neutralizing, the fat packaging, and the detoxifying all happen within minutes of a meal instead of hours. Now, the evidence calibration matters here, because not every claim about the liver sits at the same level of certainty. The glucose storage mechanism, the urea cycle, bile production, these are among the most rigorously tested pathways in all of biochemistry, settled science, replicated for close to a century now. The precise efficiency of Kupffer cell filtering under everyday conditions is harder to pin down directly in living humans, and most of what we know comes from animal models and isolated perfusion studies. So treat the scale of that filtering as well supported, but not exhaustively measured in people just yet. What makes this organ even stranger is what happens when you damage it. Surgeons have known since the 1930s that if you remove up to 70% of a healthy liver, the tissue that's left will grow back to nearly its original mass within a matter of weeks. Not by building some new structure from scratch, but by the existing hepatocytes dividing and expanding right where they are. No other solid organ in your body can do this. Your heart can't, your kidneys can't. Your liver was, in a very real sense, built to survive being eaten, and some researchers have connected that directly to the old myth of Prometheus, whose liver was devoured every day and grew back overnight. The Greeks had no way to explain the mechanism, but watching wounded animals survive, they seem to have noticed the pattern anyway. Living liver donation depends entirely on that property. A surgeon can remove roughly 60% of a healthy donor's liver, transplant that piece into someone in liver failure, and within about 6 to 8 weeks, both the remaining section in the donor and the transplanted piece in the recipient will have grown back toward a full functional mass. 2 people, 2 livers, both regrown from one original organ split in 2. That regeneration is driven by hepatocytes re-entering the cell cycle in near-perfect synchrony, guided by growth factors like hepatocyte growth factor and interleukin-6, a process first documented systematically back in 1931 by G. M. Higgins and R. M. Anderson using partial hepatectomy in rats, and confirmed in humans through decades of living donor transplant outcomes since. So go back to that sandwich. Bread, turkey, mayonnaise, swallowed without a second thought. In the 90 minutes after you ate it, something like 240 billion cells sorted its sugar into storage or straight into circulation, converted its protein's nitrogen waste into something your kidneys could clear without touching your brain, emulsified and repackaged its fat for a trip across your entire body, and filtered the bacterial debris riding along with it out of your blood before any of it got anywhere near your heart. You felt none of it, no sensation, no signal, nothing to notice at all. The most consequential chemistry happening in your body after a meal is chemistry you were never built to feel. It was never just a sandwich. It was 100 billion cells deciding, in real time, what you were about to become.